interphase is the phase that the cell grows, replicates its DNA, and grows more. So in G1, the cell grows more so that it can be large enough to split apart. In synthesis, or S phase, it duplicates its DNA, or synthesizes its DNA. And finally, in G2 phase, it grows even more. And finally, the cell is ready to do mitosis, or to split apart.
The first stage and the longest phase of mitosis is called prophase. As prophase continues, the nucleolus seems to disappear. Microtubule structures called spindle fibers form in the cytoplasm. Coming out of the centrioles are yet another type of microtubule called aster fibers, which have a star-like appearance. The whole structure, including the spindle fibers, centrioles, and aster fibers, is called the spindle apparatus.
In the second stage of mitosis metaphase, the sister chromatids are pulled by motor proteins along the spindle apparatus toward the center of the cell, and they begin to line up in the middle of the equator of the cell. In anaphase, chromatids are pulled apart. Microtubular of the spindle apparatus begin to shorten. The shortening pulls at the centrum of each sister chromatid, causing the sister chromatids to separate into two identical chromosomes.
In telophase, the chromosomes arrive at the poles and relax. They form the membrane and the nucleoli. Spindles disassemble and are recycled for the cytoskeleton.
Finally, there's cytokinesis. Towards the end of mitosis, the cells begin to rip apart. The cytoskeleton and the cytoplasm split and become two cells. Finally, the cell membrane closes, pinches together, and finally splits apart completely, creating two identical cells, or sister cells. These two cells are completely identical in DNA, and the organism and the organelles in the cells are distributed between the two.